for the fall semester in Xpert Diagnostics. Today we have two really great speakers, Drs. Dailey and Dr. Martin Jones, to talk about their work with GeneXpert at Cepheid. And just to introduce them a little bit, Dr. Pete Dailey received his Ph.D. in UC Berkeley in the microbiology department. And since then, he's worked at a number of different companies, including Chiron Corporation, Bayer Diagnostics, and Roche Molecular Systems. And from 2006 to 2011, he was the Senior Vice President of Research and Development at Cepheid, so he will know a lot about the GeneXpert system. And he is now a diagnostic consultant for infectious diseases for Ronald Harris, and he's also a lecturer at the school. Dr. Jones completed his bachelor's in biology at George Washington University and did a master's in the University of Colorado. He received his PhD in zoology and from UC Davis, where he worked on muscle contraction research. He has over 20 years of experience in the diagnostics community and is now the director of research and development. Is now a director.
The director sounds better.
Yeah. Okay, so we'll welcome you guys to get started.
Okay.
And thanks for coming.
Okay, very good. Pleasure. I'm actually Martin's. Martin's going to talk the most here. Martin actually is the person on the West Coast most responsible for the GeneXpert TB assay, and he'll tell you more about its development. And really got started, what, 2001, something like that?
Yeah.
Okay, I remember when the assay was first approved, endorsed by the WHO, and I was having, I went to the hotel and there was a board of directors meeting, and was talking to them and had drinks with them, was trying to find out what they were thinking, get ready for the board of directors meeting. And I said, you know, boy, we started working on that back in 2001. And they said, why did it take so long? But anyway, But anyway, what we're going to talk about, and I used the term nearer patient testing and avoided the use of the word point of care. And that's because I know, especially in this group, point of care diagnostics, and particularly in developing countries and work on global health in developing countries, the word point of care is used. But you will see, I think you would not find the term point of care used anywhere on the GeneXpert website, or probably on most molecular diagnostic companies' websites. So I just wanted to kind of tell you why that is before Martin starts here. What companies in the United States use particularly is CLIA complexity. And so we might refer to an assay being high complexity, so requiring a very sophisticated person to do it. And it's actually the FDA that basically judges an assay and whether it's high complexity. Most Cepheid assays, with one exception, are moderate complexity, so requiring the less technical person to run the assay because it's integrated from sample to result. And then kind of the closest thing to thinking about point of care is called CLIA-waived. And so those are assays that can be done by not a highly trained person. You have to be able to read at an 8th grade level. And these are waived from the overview by the Clinical Laboratory Improvement Act. And so that's the term that often molecular diagnostic companies like Cepheid will use that's closest to the idea of point of care. Have I got that right, Martin? So anyway, just so you know, molecular diagnostic companies don't use the term point of care. Why is that? It's because of this, because there's sort of lack of a good definition. Also, it's a little bit of a sore subject. Some of our customers are concerned about, some of the customers of molecular diagnostic companies, because they're used to controlling things, right? The big laboratories are used to controlling diagnostics, and the idea of point of care is actually threatening to them because it brings it out of their control and they have legitimate questions about quality control. Okay? So I think I'm going to leave it there. We'll, we can talk about other things if we get interested in this, and just turn it right over to Mark. Okay. Let's see. What is it? It's actually FDA. So the Cepheid mostly works with Center for Devices and Radiologic Health, CDRH. The other part of the FDA that works on that is Center of Biologics, Cepheid. But also CMS is involved, right? The Center for what is it?
Easier to say CMS.
CMS, yeah. Anyway, it's a little bit fuzzy. It's both the FDA, but also the CMS, which I think is in charge of kind of Medicare payments. So let me see if I can do some research on that. But it's actually a little. The FDA is the one that issues that ruling, and you'll usually see it when Cepheid recently had their GeneXpert MTB assay FDA cleared, right? And then about 2 or 3 weeks later comes the announcement it was as moderate complexity. It's through the FDA, but also the payers involved, so Medicare, I think, are involved in that also.
Well, it's wonderful to be here, really. It's been a long time since I've been in an academic setting. So I expect lots of different questions, lots of fresh, probing questions. He just mentioned the near-patient aspect of the TB assay. May touch on HIV and STIs, but we're working on those, let's put it that way. And this assay, just to let you know, grew out of a long collaboration with Dr. David Aland's lab on the East Coast. And we applied for grants as a collaboration. We got the money and did the work and made did publications together. So it's been a really tight joint effort in that development. And we also were fortunate enough to have, as part of this overall collaboration, the NIH injected a lot of money and helped us develop our technology that's used, certain aspects of our technology that's used in this assay. And also FIND, that's the Foundation for Innovative New Diagnostics. So they supplied some money. They're a NGO, kind of a nonprofit organization that will get a pile of money like the Gates Foundation and then find people or mainly companies that have a technology that fits into their pipeline to develop. And early on, they focused on Cepheid after doing due diligence and so forth. So it really all came together. So it was just a wonderful collaboration. And the NIH just loves us now because that was kind of a poster child for this type of industry, academic, and governmental and non-governmental entities coming together to produce this. So I'm going to start kind of, stating the obvious problems, and then where we are today, not where we will be. I'll talk about that at the very end. But I thought I would talk about where we are today first, just to give you a good idea of what we're doing right now and what we accomplished right now. And then I'll step back in time, basically, and development timelines, and pick it up from just about the very beginning. I won't touch on every technical aspect of it, several days maybe, I don't know. But just give you an idea of what we had to contend with, some of the lessons we learned. One of them being be careful what you wish for, and how it's going to impact the actual treatment and care of TB patients. So TB is the leading cause of death worldwide by bacterial infections that have high impact on HIV-associated mortality in Africa. They're obviously very susceptible to infections. TB is a dominant one in South Africa. Drug resistance, you've heard about this a lot, I'm sure, from multiple organisms. Nowhere is that more true than tuberculosis. There's now multiple drug resistant strains that are established and emerging, also humans. And there's also what we call extreme drug resistance, where just about anything you can throw at it doesn't really work well. And so this is such a key issue that it is worthy of everyone's attention. Now, drug susceptibility testing is slow and cumbersome. So this is basically you culture, you get a positive, and you take that and do some more culture. So it's weeks and weeks and weeks before you get an answer that, yes, you're drug resistant. So we have to actually, as part of the GeneXpert solution, is really shorten this. So we did 2 hours. And the magnitude of TB is overwhelming lab services. It's just a huge problem, resource limitations. These are all take place in what we call high burden developing countries. So these typically have poor infrastructure, poverty-stricken, poverty is a big deal, and just few facilities at all. Much fewer trained people. So the trained people, the big labs, tend to service part of the population. They do what they can, but there's the vast majority of the population that does not have access to a central lab. We're talking about South Africa in this case, and HPC in general. And existing diagnostic algorithms are inadequate. So this is all part of the issue that we had to deal with. And just to touch on the current TB diagnostic situation, microscopy, well, it's 120 years old now, still being used. So it's used the most often of all TB diagnostics worldwide. So you'll often have a TB lab. We call it smear. So you make a smear on a slide and look for acid fast bacilli. And so it's, I think, almost better than nothing. It's got very poor specificity and also sensitivity is not that good. So the bottom line is you miss a lot of true positives. The minority of patients go undetected for that reason. There's still long delays in diagnostics. The HIV issue is ever present, and resistance is clinically diagnosed, and that means symptomatic. So a doctor will begin treating a person for TB. They assume, based on their evidence and their knowledge and experience, that it's not drug resistant. If they fail, if the drug doesn't work, then they realize, oh, it's drug resistant. So that's what it means by clinically diagnosed. So a real fast, easy to use lab test was needed. I touched on culture already. Not only that, but it needs highly skilled staff, centralized laboratories, and from one lab to another, you'll get different proficiencies. So one lab may be really good at culture and drug susceptibility testing, but you distribute a proficiency panel to a number of labs and you'll get different results. And so there's a big issue with consistency of result. And again, no rapid method of detecting resistance. So here's what happened a couple of years ago. So this was big news. So the WHO endorsed the Xpert MTB. You see the immediate effect. Here's where the GeneXperts are or were in 2011. And there were 99 GeneXperts. There were 524 modules, by the way. We introduced the module. So this is essentially the unit scalable, so you can have multiple modules together to run separate individual assays. And so this, there was really a lot of demand, obviously, and it's growing. I'll show you a chart that shows you how it's growing. Q3 2012, about a year ago, it was about 1,000 GeneXperts. 73 as opposed to 24 countries. So people have really taken this on as a solution, potential solution to their problems with TB. Nowhere more than South Africa. The Minister of Health in South Africa said, forget smear, forget culture, it's all GeneXpert. So that was a big statement for him to make. I mean, his neck is on the line, really. But be that as it may, it's the reality. So it's had already a huge impact.
What does yellow refer to?
Well, I don't know. Those do not look like high-burden countries. So I think those are developed countries. You see Europe, the States, obviously Canada, Australia, and even I don't know my countries here. But anyway, so it's lower developed countries. Very low prevalence of TB. Not none, but very low prevalence. So here is what has happened in terms of the number of modules and the number of cartridges over several years. This starts off in Q4 of 2010 and it goes to the first quarter of this year. And you can see it started off very low, 40,000 there. And it's grown to a little over 2 million by the first quarter. This year it will top 3 million. Demand continues to increase. South Africa alone of this accounted for 1.3 million tests. That's excluding the private sector. And here are the number of modules. It's 50% of the global usage. So South Africa is something we pay a lot of attention to just for that reason. There's so much information there for us to peruse.
So are governments the main purchasers?
Sorry?
Are governments the main buyers, customers?
Yeah, so yes, there are two types of labs, public labs, and there's also private labs, and also public testing. But this is, for instance, South Africa, it excludes that private sector, so it's just the government public sector that we're talking about here. And they, for instance, they get a break in the price, a huge break actually, in the price and things like that. But, you know, South Africa really helped. There's very committed researchers there, clinicians, people that realized the magnitude of the problem and wanted to do something with it. So we worked very closely with them also. put instruments in, for beta testing. We would talk to them about their results, their experiences, what's working, what's not working, what problems are you having with the data, the results, and so forth. So they were very, very connected with the company actually during the development. So that's where we are, and this is going to keep trending upwards. This has been, the growth is amazing. So the impact on the company has been amazing, because here we can get away with being sort of a low volume manufacturer. Now, it's like the sorcerer's apprentice, right? So we have to become a high volume producer, producing high quality materials. No change from the quality here, we're just going to do lots more of it. So it's really a challenge. We're meeting that and investing in the right methods and personnel to make this happen. So, this is the Minister of Health in South Africa. This is the guy whose neck is on the line. This is how simple it is to run the test. So when I say simple, like anyone in this room could run the test. If I show you for 10 minutes, anybody take a sample, put the sample in the cartridge into the machine, and press a button. Who can't do that? So it's that easy. And so you're combining sample prep, real-time PCR, amplification, and detection, all in one little cartridge machine. So that's what it means by simple to use.
Sorry, is it sputum sample?
Yes, we are looking at other samples. We've tested over the years CSF. We're working on stool for pediatric patients. Every so often it's used off-label. It's not used for nursing. It's used for sometimes tissue biopsies and just things people want to try and see the result. But it's made, yes, for sputum. First design goal was it's got to work with the primary specimen. So there can't be any involved processing of the sample by the user. And if that's satisfied, then we cover all the different cases. Good job. Yeah?
Can I speak up?
Oh yeah, sorry. Did you hear back there? Yeah, I should speak up, just tell me. That's what I tell everybody else that I listen to. Speak up, please. Okay. So now we're going to step back a little and then catch up with ourselves and some more slides. So the company was founded in 1996. Someone's living room. It's not a garage. It was a living room. It's in Sunnyvale, Silicon Valley. Lots of smart people are there, and the founders were from microengineering, lab on a chip people. The founder, Kurt Peterson, invented and made the accelerometer that goes in the air bag to the plant. So it's a tiny little silicon chip with a detector. It moves in response to sudden deceleration. So that's what is in all those cars. And biomedical industries. We had funding sources early on, so the U.S. government, Department of Defense, their worry was bio-warfare agents. So we did a lot of work with that early on. There were some private investors and then the public in June of 2000. And I think it's important to mention the central theme of Cepheid. So what is the vision? The initial vision is so important that you're going to develop it and you're going to build on it, build from it. So the vision is there. It's a valid, real vision like you would anticipate having for like an apple or something. So it's, what is it? What is your company going to be based on? Sample prep, folks. In this machine, here's the actual made mock-up, or really drawing of the early machine, sorry. And that little thing looks like a yogurt cup. That's the a facsimile of a cartridge. So sample prep, amplification, and detection in less than 30 minutes. That's the vision. And all the details, obviously, you can say right away, well, what about this? What about that? That comes later, but you got to build on this and move forward with this. And the vision could crumble. You know, it could say, well, we're not going to do this anymore. We're going to get into the sequencing. But it didn't. It really worked. And the company has grown because of this simple vision. I'm not saying we achieved this, but that's the vision. So it's got to be simple, obviously, and so forth, and rapid. And here is what I think is a very telling chart. So this is what I think really changed the way Cepheid thought of itself early on. I mentioned Kurt Peterson and his silicon lab on a chip ideas, and nanotechnology, etc. We were involved in that early on, and it became apparent after looking at something like this. So this looks at sample volume versus sensitivity. You can see there's three types of diagnostic tests here. Clinical chemistry, blood, urine markers, serum markers. Immunoassay, antigens and antibodies, looking at those. And down here in this realm, far lower right corner, DNA probe assays, PCR assays. So what you see is the sensitivity range here. This line is a theoretical line. So on this side of it, perfect detection and statistical confidence in the result. This dotted line, also highly theoretical, is less than 1 molecule per sample in the realm. And what this means is, if you're looking for this many, if your sensitivity is this many, then based on the markers you're looking at, you can use very, very tiny volumes, nanoliters. You can do the lab on a chip. Immunoassays, less so. So you see the range for immunoassays is still encompasses nanoliters. But you know, it's very typically you'll take single microliters or 5 microliters for an immunoassay. But you get down here to where your sensitivity, your capabilities are certainly below 10 per mil. 10 copies per mil, 10 organisms per mil. How do you get that? What's present in the biological sample? And you see, well, there's milliliters, or tens of milliliters necessary for very low concentration samples. For instance, in sepsis, 1 CFU per mil is very important. How are you going to detect 1 CFU per mil with a nanoliter? You're going to have to get horrendously lucky, right? It'll almost certainly not be the alibi you take for testing. So what you have to do then is figure out how do you process larger volumes. Milliliters, sample. Not microliters, but nanoliters. We have to get up to the milliliter scale. And lo and behold, that's where the cartridge idea came. So if you can imagine a very dilute sample of a certain volume, say 10 mils, you can, I'm sure we've all used syringe filters. So the idea is force all of the sample through the syringe filter. And the syringe filter has a pore size smaller than the organisms you're trying to capture. The organisms are retained, everything else goes through. You're left with a concentrated sample. So that's the whole idea. That's where it all started. And this is the original version of this cartridge. So this is a disposable. It's a one-use cartridge. You use it, you never open it after you put the sample in, and you dispose of it correctly after you're done. We started off, we had to start with machined acrylic. It actually screwed together. We actually had to bleach it in between runs. We had to reuse it. You know, they would have killed us if we started throwing these away. We expect me to make more of these, you know. So that's where it started. It didn't start like with the final implementation of the final design. You got to take little steps along the way. And this is now entirely plastic. This molecular biology lab is plastic. It's injection molded, all the parts are. And it's assembled, it's very simple to assemble. And like I said, you throw it away when you're done. So that was sort of the beginning. And along the way, this is what we developed. So in order to make the concept work, the concept of filtration capture, we have to actually lyse the material. So these are intact organisms. You have to get at the DNA. You have to break open the organism somehow. So we did ultrasonic, we used ultrasonic energy, ballistic lysis, to break open the organisms. And so within this little part of the valve body where the sample was captured, we have a lot of silicon glass, very small silicon glass beads. So that's the ballistic part of the lysis. So the ultrasonic energy will cause those beads to fly around and break open your organisms. So this is the critical interface between the device here and what goes on inside the PCR amplification and detection. Cold chain. We don't want a cold chain. A cold chain is where everything has to be refrigerated, kept in a freezer. You've used polymerases, you've used all kinds of things in the lab. What do you do when you use a polymerase? Pull it out of the freezer. And you use a tiny bit of it. And then you put it back in the freezer. We couldn't do that on this. So we had to develop a whole technology around lyophilization. And not only just lyophilization, but lyophilization in little spheres, like a BB. Just imagine a BB. And here's a little vial for afterwards. You can check it out. And I mentioned, after you put the sample in, you don't open it at all. So it's all sealed. So cross-contamination, contamination by amplifies is horrendous to get rid of. We had an incident early on. It wasn't our fault. But it was so tough to get rid of. We got into the reagents, and blood negatives. No, negatives are all positive. So people are very, very sensitive about this. That was one of the knocks of doing molecular biology in the clinical setting, PCR amplification. They went to great lengths to prevent contamination, negative air pressure rooms. You had to build rooms, specialized rooms in order just to do the testing. Not so with this. It's enclosed. We've done tests. Does the GeneXpert generate any aerosols, either infectious or amplifies? The answer is no. We did extensive studies. Nested PCR. So nested means there are two primers that we call the outer primers, and then that's part of the first PCR reaction. The second PCR reaction is actually run on two what we call inner primers. So that gives you a nested approach. And you can imagine the increase in amplification. So you're taking the first amplified product, which is longer, which there's probably a lot of already, and then you're re-amplifying it with inner primers. So you have a shorter amplicon. It happens, and second amplification operates more robustly and quicker than amplifying a larger stretch. So we can do that with the cartridge. In fact, we do that with the TB assay. Built-in assay controls are key. There's a thing called erroneous but believable results, EBBR. So that means something happened during the run that caused an error, but the system or the assay didn't alert the user that something had happened that would potentially affect the result. So there could have been a missing reagent, for instance. What if one of the beads didn't get into the cartridge? Well, you get a negative, but you wouldn't know it unless there was a specific control for that. So we spent a lot of time with making redundant controls, really. The hardware, all the optics, fluidic drives, etc. It's all a redundant system looking for errors each and every run. Universal sample prep. So sputum, I mentioned, obviously is a TB sample, but also stool, blood, bronchiolar, alveolar lavage, and swabs. So for a flu test, the swab, stick it in the cartridge. It's that simple. Here's the module. I've already shown you this module. And so we can have a little blow-up of the module here to look at later. This is like a plastic version of the uniframe. Here's all the components that actually come together in that module. And drive the assay forward. So it's got syringe drive for fluid movement. So there's a rod that comes down, moves up and down, and acts as a little syringe. You'll see it's much easier to see once you get up there. There's a valve motor on the bottom that directs access to chambers, so it turns the bottom of the cartridge, which is a valve body. It has ports in it, so it has random access to any two chambers. So it can transfer material from one chamber to another, either directly, that means not through the filter, or it can transfer through the filter if you want to filter something. If you want to wash the sample you've already captured, you can do that also. Their independent thermal cycler allows for random access design. This is self-contained. It's got the thermal cycler built in. It's got the optics built in. Everything's right here. And that makes it a modular system, and that makes it scalable. So we have actual GeneXperts that are one. I don't know if anybody buys it, but it's this big. It's barely larger than that. And we also have up to 80 modules in the system. We call that the Infinity. It's much fancier. But it's automatic loading and some robotics thrown in to move cartridges back and forth. How it works. This was such a neat little video I can't show you because the file is in the wrong format for this computer. But it's on YouTube. We're on YouTube. So just look at, for Cepheid, really, and you'll see this as one of the videos on YouTube. It's really worth looking at just to see how this works for a nested assay. So you'll just have to imagine it for now. It's very important to identify your target, obviously. So the goals were detect TB case detection, and also determine if there are mutations in this certain hot zone of the RPOB gene. So these little stars or asterisks mark potential sites of mutations that are possible. So this is 81 base pairs. And the idea is to detect mutations anywhere along this stretch. And so how do you do that? Oh, there it is. So why this section? Well, rifampicin is one of the first, it's a major first-line drug. And if you have a mutation, it becomes rifampicin resistant, it's a really good marker for multiple drug-resistant TB. Also, what about the U.S.? Here we get into the differences between low and high prevalence. Low prevalence, a mutation might not mean an actual rifampicin resistant organism. There are silent mutations. And here is what the real-time amplification growth curves look like. So the end user in a clinical lab would never see this, they'd just see the answer. We have 5 molecular beacons, that's a type of probe, each one labeled with a different fluorophore. It's quenched when it snaps together. So that means it's dark, unless there's a specific target there. If there's a mutation, and here you see all 5 come up. So that's TB detected, rifampicin sensitive. It's a wild type. Here's our internal control here, depicted here. But if there's a rifampicin resistant mutation, a mutation in that 81 base pair zone, you see there's only 4. It's hard to see, sorry. So 4 probes have come up, and there's one that's a flatliner. So there was a mutation there. So it remained dark basically throughout the whole amplification process. So the software can interpret this and flag it as rifampicin resistant. So that's really the whole assay right there in terms of PCR, real-time PCR.
So just one mutation sequence will trigger the disease-based system.
Sorry, I'm sorry.
So do you need a combination of mutation detection or just one?
Well, for rifampicin resistance, just one would do it. That often does. So there's certain mutations that are very, very prevalent in rifampicin resistance cases. So they account for maybe 40-something% of the worldwide resistant cases. In fact, some assays are based on that. They'll look at the top 5 mutations, because that's 90% of the total. You still have the 10% that we want to detect.
So why, I don't kind of understand, why do you have 5 curves there? Is that essentially, or 4 curves essentially tell you the number of, it's only sensitive to 4 mutations then?
Okay, imagine this was wild type. That means there's no mutations. It's rifampicin sensitive. We want to be able to detect a mutation anywhere along that. So thanks for bringing that up. It's a key piece of the design of the assay. The beacons actually individually span a given region of this. So it takes 5 to span the whole region. So that means in order for it to be a TB positive, a certain number of probes will have to be positive. Rifampicin resistant, certain number of probes have to be positive, but a certain number, at least one, have to be negative. So if you have a mutation here and nowhere else, that will affect the hybridization of just that probe. So one of the probes, sometimes it's several, the signal will become much weaker, or it'll drop out altogether, like you saw in this picture. Does that explain it?
Yeah.
The real world challenge. Nanotechnology, this, basically. So, okay, we gotta look at these samples and somehow detect TB in them. This is your basic sputum. Number of puns, you can think of easily a number of puns. But here are some more samples arrayed along the bottom. So these are just your run-of-the-mill. This is what you're going to see. In fact, even worse. Be bloody, purulent, all of the above. The viscosity varies widely. Watery. Here's just a sample from some field data during the early days. Out of 100, and the intake nurse, noted the consistency of the sputum. 17, very low amount in total, was watery. Viscous, 38 out of 100. That means it's thicker than water, but it still flows, When you tilt the cup, it'll move. And then very viscous, the large majority of the samples. So very viscous means you tilt the cup, it barely moves or doesn't. Some of it's semi-solid, so it's gravity defying. If you turn the cup upside down, it won't go anywhere. How do you work with that? They do a diagnostic, sophisticated diagnostic, PCR and stuff. Look at sputum processing. There are chemicals you can add to sputum to make it liquid, for one thing, and to also inactivate the TB in addition. So this is common, is to use sodium hydroxide and N-acetylcysteine in a method to liquefy the sputum and prepare it for further testing like culture or smear microscopy. Typically, though, this is what you do. You add the chemicals, you incubate it for X minutes, 15, 20 minutes, I think. You have to spin down the sample, make a pellet. You don't want this in the final reaction, so you have to get rid of it. You then wash the sample, that means resuspending it, recentrifuging it. And then you resuspend finally in a mill buffer. There's batch dependence on this and subsequent steps. You don't want to do just one of them at a time. So you wait until you've collected enough to run a batch. So with that batch, typically for molecular diagnostics, you'll extract the DNA from it. And you'll use, of that, only 5 or 10 microliters per PCR. And so this results in a time delay, obviously due to batch, dilution, and subsequent loss of sensitivity. So our approach was to essentially do this. We used different chemicals that were much more effective than the standard chemicals. They include sodium hydroxide and isopropanol. That's part of the chemicals in there. It will not only liquefy the sample, but it will inactivate the TB. One of the requirements, the design requirements for handling TB outside the hood. You don't want your GeneXpert inside a ESL3 hood and try and use it in there. So you want to use it on a lab bench somewhere, out in the open lab. And so you have to inactivate the sample. So we spend a lot of time perfecting the ability of our sample treatment reagent to prepare the sample for actually analysis. So this is one time where out of the cartridge processing, some handling was necessary. But guess what? It's still moderately complex. So, and you talk to some experts now and they say, wow, what you guys did was enable the sample to be analyzed. They don't care about the rest of it, but this was really a big deal to get this solved. And so just touch on some controls of the system, alert the user to errors. I mentioned erroneous but believable results. They can arise from a variety of conditions, missing reagents, poor reagents, degraded reagents, the system not heating correctly, not moving the syringe up and down correctly, all those. So what we want to do is we do failure mode defects criticality analyses, which you're familiar with that. And so we drill down to the causes of all possible EBDRs. And we need to get these, oh, there it is, sorry. We need to get the EBDR rate very low, close to zero. So system features that allow this control is real-time fluorescent analysis in a sealed tube, no contamination, no wet interface between the instrument and the cartridge. The plunger rod that comes down only contacts the plunger that's already in the cartridge. So it never touches wet solution unless something catastrophic happens. Total internal control of the reagent system. So you don't have to run external positive and negative controls like most, not all assays. And then there's a system monitoring for the quality of the electronics, the hardware, software, thermal, optical, etc. So that's what is present each time you run a GeneXpert. Internal control, so this is critical. It's co-amplified with targets. What happens if TB is not present and you don't have an internal control? You'll get nothing. Did your reaction work? Did you filter the organisms okay? Did something happen? Why did you get no result? So this internal control, you can also call an internal positive control. So under normal conditions, this internal control, which you identify because it's got a different fluorescent dye on it, should be positive. So if it's positive, that means everything worked, there's just no TB there. So it's a negative. So it's a valid negative. If the internal control doesn't come up, TB doesn't come up, it's an invalid. You've got to repeat the run, go back to the patient, etc. It can be used for quantitative normalization. We can use controls that's already in the sample, or we can add it during manufacture. It can be competed out by a positive sample, so if it's a very high positive, it'll monopolize the PCR reaction, and the control will be negative, but that's okay. Because you have positive signals, so you know everything worked. And so we see it can respond to sample inhibitors. That's another big issue we deal with in all types of biological samples are inhibitors. So these could vary widely. So heme in blood, for instance, is a very potent inhibitor. And so when you listen to people in the TB world, they talk about point of care, Now, anything that's any decentralized testing, like we want the GeneXpert to be. GeneXpert can be put into very rudimentary spots. It has to have power. That's its own basic own requirement. And they say, high tech. Why is high tech not suitable? They're thinking of the high tech lab, the multiple rooms, the technicians running them. But based on what you've seen, I think you can agree this is high tech that's used in a low tech environment without trained personnel present. And so we've done all the hard work for the user, the end user. We've made it so they can reasonably, with confidence, do a good run over and over again. And here's where it ended up. So this is out of a publication. So this is all you do. This is our sample treatment reagent. You add that semi-quantitatively. You can eyeball it. It has to be, for instance, 2 to 1, 3 to 1. And you mix it, let it set for 15 minutes, and activate TB. Transfer 2 mils of that into your cartridge. And you put it in the machine. That's the end of hands-on work. The machine does the rest. So it's what they call walk away. You can just walk away now. You don't have to do any manipulations, any further manipulations. And what happens in the instrument, just to recap, the sample is automatically filtered and washed. The ultrasonic lysis operates on the filter captured organisms to release DNA. You mix the DNA molecules with the dry PCR reagents. Remember that reagent beads are in there, just waiting for the sample. So you move the sample into the chamber with the PCR reaction beads or reagents and mix them together. And then you do your nested real-time amplification and detection. That's it. And this is what the end user would see. Just very simple. What's your test result? MTB detected. Rifampicin resistance not detected. So it's sensitive to rifampicin. The back of this result are all the analyses and workflows, etc. And I mentioned FIND already, and people in South Africa, but also for running clinical trials. How do you know your assay works? How do you know the design is a good design and you've done all your homework and your thorough development? You have to test it in the field. So with the collaboration of FIND, that was relatively straightforward because they had collaborations previously with all of these labs. They knew these people intimately. These people are lab members here, very interested in this type of diagnostic. So Peru received HIV, 2% prevalence, culture positive TB, 61%, drug-resistant TB, multiple drug-resistant TB, 7%. And you look at the different populations and you realize that some look like they're in real trouble. India, this is a tertiary care hospital. So TB is 60% of all the samples. Drug resistance, 67% of hospitals. So this is, just a dire situation. This type of environment. TB and HIV, deadly combination. You have to diagnose it. If the patient is to have any chance at all of survival, you have to diagnose it rapidly, not get the result in 8 weeks. You need the result now, and you need to start therapy now. So here is, based on the testing from the labs I just showed you, here are the results from all that testing. Greater than 1,700 patients, many more samples than that. There were multiple samples per patient. We submitted through with FIND and the David Land lab, and all the principal investigators from all the labs. An article to New England Journal of Medicine was accepted. So that was big news. That means we did our homework, basically. Doesn't mean homework ended. However, you still have to keep on top of it. But you see, smear positive patients, GeneXpert, 98.2% sensitivity. Smear positive means you look at it through a microscope and you can see organisms if you stain them correctly. The sensitivity for that, depending on the lab and who you talk to, is 5,000 to 10,000 CFUs per ml. So I'm thinking we better detect those, and we did in almost all cases. Some of the positives may in fact have been non-specific. You see a little dot down there that's stained. Is that really TB? You don't know sometimes. So you have to do a confirmation test. The specificity, 99.2%. This caught people's eyes, I'm telling you. And then smear negative, notoriously difficult to detect. They can be smear negative, you can't see it with a microscope, but if you grow it up, it turns positive to culture, either solid or liquid. Even with those, 90.2% sensitivity with 3 samples. Astounding. You get that kind of sensitivity in smear negative samples. If we just use one of the samples from the patient, 72.5% sensitivity. That means smear did not detect these. So we tested smear, since these are all smear negative, 72.5%. Patients with rifampicin resistance. So if you look at the positives, and you look at rifampicin resistance compared to drug susceptibility testing, the gold standard, very respectable sensitivity and specificity. So that's what you're looking for. The challenge now is to make sure that every month, every year from now on, the test performs like this. It's a whole different talk about manufacturing. We want to decentralize this test. You know, when we talk about democratization of diagnostics, GeneXpert's perfect for this. There's a mobile lab. It's from the Fraunhofer Institute website. It's in Africa. This is one of the slum areas, probably near Cape Town or Durban. It contains a GeneXpert, so here it is. And it's equipped with biological cabinets and biological safety cabinets and so forth. Here's the cartridge being scanned. It's got a 2D barcode on it now. And it's going to run. So this, how great is that? The patients, if you think about it, poverty-stricken patients, how are they going to even get to a central lab or central clinic? A lot of them walk. If they walk, they're away from home and work for maybe days at a time. Those days are totally lost to just waiting or trying to get diagnosed. This is so great. So in summary, for this part of the talk, for lab and the other applications, the system is designed to process a single sample as a run. So one cartridge, one use. The beacons probes can be pre-assay QC directly in the specimen reaction mixture. So while they're fluorescent status in the reaction mixture, you know if they're good or not. The internal control system has been designed and added. External negative control not required. No user intervention in processing or interpretation of results. And looking forward now, conclusions. Next generation platform technology. You can see this maximizes medical impact. So key, stay closer to the patient. It's on demand. You don't have to wait to batch your samples. Turnaround time is shortened. We're doing other assays. The system connectivity enhances the value of decentralized testing, what I'm going to show you next, by employing advanced data reporting capabilities. So people at Cepheid are very keen. They don't just rest on their laurels. They're always looking to make things better, to do different things. I would have never thought about this, but this is really great. The democratization of molecular testing is here. So what we see here is real-time test data aggregation in South Africa. So you can see these symbols here are different places where the GeneXperts are located in South Africa. So what happens is a lab will de-identify the test data. That means you scrub the patient's name or identification from the result that you're transmitting the Amazon cloud. So you put that data on the Amazon cloud, and from that data, with the correct software and databases, obviously, you get the TB rates. You know, just look at these graphs that are possible, reports by week, the number of TB resistance cases over this whole area. It's amazing. And any instrument or user problems will flag errors. And here we see at a given hospital, 17.65% TB positives out of the samples they tested. That's so key. There could be an outbreak of a given strain of TB, a given drug-resistant strain, for instance, and you could track it back with the correct data to the index case. Where did it start? How did it spread? So that's all in this approach. Now we're going to move on to the super secret part of the talk. So we're going to talk about